ways that pressure drops.
What's pressure drop?
Okay, so like I said, remember I just said if you stand up and pressure drops, then those afferent arterioles will dilate and allow more blood to enter at a given rate to maintain the flow.
When we sit or stand, that affects our kidneys?
Pardon?
Or sitting and standing affects our kidneys?
Yeah, it affects overall blood pressure. And overall blood pressure is reflected by the blood pressure in the renal artery and in the afferent arterioles. So exercise raises blood pressure. The different things,
So
volume
standing
and
for a long
stuff.
period of time actually will raise your blood.
Pardon?
Standing for a long period of time actually will raise it.
No, transitional, standing up or sitting down, you know, because normally we have tonal effects in our lower limbs that help to return that. Okay, so then the tubuloglomerular effect is involving our juxtaglomerular apparatus, which contains primarily two types of cells. So those two types of cells, it's confusing because juxtaglomerular is used both in the overall name of the apparatus and with the cell types. So the juxtaglomerular apparatus includes smooth muscle cells of the afferent arteriole that are known as juxtaglomerular cells. Juxta meaning next to. So juxtaglomerular cells are smooth muscle cells of the afferent arteriole. Specifically the ones that contain granules of renin. Because of those granules, another name for these cells are granular cells.
Oh, there they are.
So that might be easier than remembering juxtaglomerular apparatus versus juxtaglomerular cells. Okay. Now renin, I didn't go into very thoroughly when we were talking about, where's my picture? There we go. So here's those granules of renin within the smooth muscle cells. Okay. On Thursday, we'll come back and talk about this in more detail. I know I keep promising this from the heart to this very quickly, so you wonder why I'm not dropping the word and moving on. Renin is an enzyme that converts a blood pressure molecule known as angiotensinogen. Angiotensinogen is made by the liver. The enzyme renin converts it to angiotensin 1. Angiotensin 1 in the lung is converted to angiotensin 2, and it is a vasoconstrictor. So it's going to constrict arterioles all over the body. It also increases the release of antidiuretic hormone and aldosterone, primarily antidiuretic hormone. All of those, as we learned with the heart, increase blood pressure. So when blood pressure drops in the afferent arteriole, one of the responses is the smooth muscle cell releases renin. In the blood of the afferent arteriole, that then begins a sequence which is going to end up increasing systemic pressure. Okay? Through all of the, well actually, when blood pressure here is I misspoke, when blood pressure here is low, renin is released and ends up increasing overall pressure. So if blood flow into the glomerular capillaries is low, and glomerular filtrate pressure is low, renin is increased. Okay? So let's go back to this slide and look at the three mechanisms that increase it. And again, we're going to look at this again on Thursday. So low blood pressure, the macula densa cells, or even the sympathetic nerves themselves can cause these muscle cells to make more renin, all in response to low systemic pressure. Okay? Again, I'm just going to leave it at that. We're not going to go through the whole process, but I do want to talk about the macula densa, because the macula densa cells are the other component of our juxtaglomerular apparatus. So these are the granular cells, or JG cells. The other component are these cells that you see here facing the capsule, and these cells are known as the macula densa. Macula meaning spot, and densa meaning dense. So because those nuclei are so close together, they form a dense spot. So these are cells of the distal tubule. They are sensitive to the concentration of sodium ions in the filtrate. So for a very general effect,
Sensitive to sodium or just the
just
macula
the
densa?
macula densa cells. Okay? So right here. So very briefly, and I know I'm not trying to tease you, I'm talking about this and we're looking at what regulates glomerular filtrate. But we haven't talked about how the glomerular filtrate, what happens to it before it comes back to the distal tubule. So the filtrate is formed. A high flow rate is going to form a high pressure, and more filtrate is going to be formed. And it's going to move through the nephron more rapidly. A low filtration rate is going to be low pressure, but it's going to flow through the nephron more slowly. So if pressure is low in the afferent arteriole, we already know that we get renin secretion, and changes occur that increase pressure flow, all right? If that pressure is low, the filtration formation is low, and as it moves through the nephron, it moves more slowly, and there's more time to take out sodium ions. Okay? So when that filtrate gets back up, comes back up at the distal tubule, it's going to have fewer sodium ions than normal. The macula densa are sensitive to that, and they send a signal to the afferent arteriole. They release prostaglandins, and that causes changes in the afferent arteriole. All right, causes it to, and that changes flow rate.
Which cells are the granular ones, the orange ones?
Yes. So these would be the granular cells right here.
Which are also smooth muscle?
Which are also smooth muscle cells. Okay, so right now I just want you to recognize that there is that relationship between the afferent arterial granular cells, the macula densa, the flow rate, and then when we get to regulation of fluid and electrolyte balance late on Thursday, we'll come back and cover that.
I'm just thinking about this, lack of sodium, more flow. Okay. I wanna make sure.
Okay, so there are some slides in here that I put in last night, well, some of these are already here, that I will go back over again on Thursday. I just wanted to introduce you to the concept and to these structures. So when we look at some of our models of the kidney, we have two different forms of this particular model. So here is our afferent arteriole, and this is representing the granular cells in the wall of the afferent arteriole. Here is the macula densa, and these cells then are the ones that are the distal tubule that are sensitive to sodium ion concentration. Okay? The other model that looks like this, they don't show the macula densa cut, but you can see it on this particular model. And I'll show you the macula densa on the light microscope as well. So here again is our capsule. And here's our distal tubule. Notice how it changes and all those nuclei get clustered tightly together right as it comes close to the glomerulus. And so this is the macula densa right here.
That's the idea is.
That packed group of cells right at that
what's
region.
this?
Okay? So for right now, just understand that the juxtaglomerular apparatus is a group of structures between the afferent arteriole and the distal tubule that are capable of changing, modifying the flow rate through the capsule to adjust either up or down the amount of filtrate that's being produced. And we'll come back and talk about other forms of that. Okay, let's stop here and take a break. And when we come back, we'll talk about what's happening in the proximal convoluted tubule.
So, high sodium diet.